Hello, everybody. Thanks for sticking around. You can smell the wafting food, so we'll try to keep it punchy and on point. So, Invion, we have a technology that is a combination of a drug plus light. It's called photodynamic therapy. We're primarily focused on treating multiple cancers, and we also have a technology, the same technology that works in infectious diseases. We're at clinical trial stage. We have a phase 2 prostate cancer trial that was completed last year. We're currently recruiting in our second cancer type, skin cancer in Australia, phase 1 and 2. And we're making preparations for a rare cancer a clinical trial with Peter MacCallum Cancer Centre. And so we're truly in the clinic now. In addition to working with Peter Mac and also Hudson Institute of Medical Research, this past year, we've started working with industry who are co-founding some of our research programs, and I'll talk to them a little bit later. Long story short, last year we were basically curing cancer in mice. We are now focusing on human beings, and that's the stage we're at now. On the right-hand side, what you can see is a number of catalysts over the coming year plus. The dark green on the two top triangles are essentially clinical programs that we are funding, i.e. a phase 1 and 2 skin cancer program, as well as an anogenital cancer program, again with Peter Mac. The bottom 4 light green catalysts are essentially ones that aren't costing us money or someone else is paying for or doesn't cost, very limited resources from our side. And one of them is a Korean pharmaceutical company called Hanlim Pharma, funding a preclinical program in esophageal and glioblastoma cancers. And there's another Korean company that is funding a clinical program in HPV virus. So how does our technology work? We essentially have a drug that is derived actually from chlorophyll with a few extra processes. And by itself, it's very safe and really inert. When you shine a frequency of light, it generates an oxidation process that kills just the cancer cells it's localized in. The question then is, why does it just localize in cancer cells and not healthy cells? We're taking advantage of something called a Warburg effect. Cancers like sugars. And so what we did was we coated our core molecule with glucose conjugates. As a result, multiple cancers will actually be attracted to this compound and gobble it up. And then when you shine that frequency of light, it generates that oxidation process which kills those cancers. That's the first mechanism of action. The second mechanism is you've got bits of dead cancer cells floating around, makes it easier for the immune response, the immune systems to start identifying and fighting the cancers. We have a large number of novel and patent-protected compounds. In fact, we've got over 400 now, which we developed largely during COVID. We chose one of them called INV043 to be our lead cancer candidate. And we actually tested on many cancer types in vivo in animals. So pancreatic, T-cell lymphoma, triple-negative breast cancer, anal and ovarian cancers. It regresses all of those quite consistently. It's very potent, much more potent than other photosensitizers that have been approved in the market. But the flip side is it's extremely safe. We actually have applied it IV at 100 times the therapeutic dose, and I haven't found any major toxicity issues. But the key issue here is, how does it work in humans? And last year, we completed an investigator-led phase 2 trial in prostate cancer. And it was essentially a trial that involved men who were in their 60s on average that had intermediate prostate cancer. What they did was they had a liquid with our drug in it. They swirled it under their tongue and was administered systemically, what's called sublingually, which got into the bloodstream. It was an outpatient procedure. They came back the next day It's given time for the drug to localize in the cancer cells. You shine a light up the place where light doesn't shine, and that kills the cancer cells alone and left the healthy tissue intact. What did we find? From a safety perspective, we found it was extremely safe. In fact, the most common side effect was fatigue, just being tired. If you compare that with standard of care, chemotherapy, radiotherapy, surgery, there's some pretty devastating side effects that men have to go through that. That's safety. What about efficacy? So we actually measured two efficacy endpoints. One was a physical endpoint, which was around, did the tumor get smaller? That's a RESIST framework. It's an internationally recognized framework. We found about a 40% response rate. The second was a chemical biomarker called the PSMA PET test, and we found a roughly 44% response rate. Now, these are pretty interesting efficacy signals, very compelling safety signals, and so we're looking forward to, I guess, the next stage of the prostate cancer program. But what we did want to do was to show that the same drug and the same treatment can be used for very different cancers in different formulations. And that's why we're currently recruiting in a phase 1 and 2 non-melanoma skin cancer trial using a topical formulation of the ointment in the world's capital of skin cancer, which is Australia. 2/3 of every Australian will be diagnosed with skin cancer by the age of 70. So there's a very long need there that's not going away. The other reason we're focused on skin cancer is because it's an ointment. It doesn't get into the bloodstream. Regulators feel more comfortable. The trials are cheaper. It's a faster pathway to commercialization. And we can get the same ointment formulation used in our anogenital cancer trial, and we can piggyback off the data from this skin cancer trial. In terms of the market need we're looking to address, skin cancers are now typically treated with surgical interventions, which you see on the bottom right-hand side. And typically they work pretty well, but the scarring can be pretty nasty. There's a second type of treatment, a very old version of PDT, which actually addresses the scarring, but doesn't work as well and can be extremely painful. What we have is something we believe has the potential to work equivalently or better than surgery, certainly have equivalent or better cosmetic results, and we have not noticed any pain in any of our previous studies. So long story short, we potentially have a treatment for skin cancer where it doesn't leave scarring and it also doesn't cause pain. That's where we're trying to potentially displace the current market or parts of it. Now, our bigger picture plan is to find the same drug to treat multiple cancers. And in fact, there's a second wavelength of light that activates this compound. Instead of killing the cancer cells, it actually fluoresces them. That's what you're seeing on the left-hand side. This is a picture of a mouse that had cancer. We injected the drug into the organ, waited half an hour. We got a light that you can buy for $10 on Taobao, and we used an iPhone to take that photo. Very clear. You can see at a cellular level, the pink is where the cancers are, including little metastatic spots. So what that creates is potential pathways towards both therapeutic as well as diagnostic applications. And that's the predictable stuff. The holy grail stuff is if we can actually activate the immune system so that the immune system starts fighting the cancers directly. And we did this study with the Hudson Institute of Medical Research with immune competent mice. They were implanted with triple-negative breast cancer tumors, which are typically chemotherapy resistant and typically spread or metastasize. You can see in the bottom two panels, the mouse treated there had full regression of the original tumor lesion, no scarring, which is relevant for the skin cancer trial, and no sign of metastases. The second, I think, more interesting element of this study was about a week later, we tried to re-implant the tumor into the mouse and it would not grow. And we found from immune work the mouse's immune system had started to identify the cancer and actually started fighting it without any further intervention. Speaking of immune systems and immunotherapy, that is the blockbuster drug in big pharma today in cancer. And typically the blockbuster drugs are called checkpoint inhibitors. They basically stop the immune system, from the cancers from hiding from our immune system. When they work, they're great, but they normally don't work. So the response rate's typically 12%. We improved that to 80% in our preclinical models with Peter MacCallum Cancer Center. So if we can translate that to humans, that could have quite dramatic impact on the efficacy of immunotherapies today. So if you take a step back, we essentially have a portfolio of compounds, and we already have a portfolio of clinical stage assets targeting multiple cancer indications. We also have another drug candidate that's targeting a totally different disease area, which is infectious disease, specifically HPV virus, that another company is funding for us. And we have not given any rights on top of that. The last area I'd really like to look on is a very different view on the addressable market. The average, most drugs, as you know, innovative drugs are developed in the US. The average cost of a new drug to patients last year with the FDA was $300,000 US. I don't know many Americans that can afford that. And certainly in the rest of the world, I think less than 1% of the world can afford that price tag. That's because there's a lot of movement towards personalized medicine and very sophisticated technology that's very personalized to the patient. We have a small molecule that works, that's very scalable in manufacturing. The same small molecule can also be treating multiple cancers. And so we have the capacity to basically make this much more affordable, which means that no matter what the efficacy and safety profile of these new drugs that come in, they're just not affordable by 99% of the world. We hope to bring that to top 10, top 20, top 30% of the world, which means 10, 20, 30x impact an addressable market that will never be able to afford those new treatments. So we think that's another dimension of what we're looking at. I think I'm going to stop there because I think we're probably close to time, but we're in booth 34. If you want to have a chat, feel free to come over the next 2 days. I thank you very much for your time and hope you enjoy lunch.